Inside these museum cabinets are time machines to the past, specimens collected by naturalists over hundreds of years. By unlocking the information coded in its genome, the little toe on this bird can tell us a century of history. These birds were collected nearly 100 years ago when Field Museum scientists made an expedition to Ethiopia. Like on most museum expeditions, the scientists documented the diversity in the region and collected specimens. These specimens were immediately used by scientists to better understand species diversity and distributions. Since then, they've been stored in the museum's research collection, and now we're using them in ways the original collectors never could have imagined, to collect genomes. I'm Amie. I'm a researcher at the Field Museum. We're currently using some of the century-old specimens from the Ethiopian expedition for new research. I'm collaborating with scientists here and at Texas Tech University to investigate how forest birds in the Ethiopian highlands have fared over the past century amid drastic changes to the landscape. The Ethiopian landscape comprises grassy highlands and forested mountains, dissected by drier lowlands and valleys. These diverse habitats support a large number of species. This, in combination with global increases in human development in the past century and the resulting habitat change, makes the Ethiopian highlands a region of conservation concern. To learn how these birds have fared over the past century, we're collecting and comparing genomes from 8 species. Some genomes from old museum specimens I told you about earlier, and some genomes from birds alive today. To collect genomes, we first take samples. For the modern, live birds, collaborators took a small blood sample. For the old museum specimens, I cut a small piece of dried skin from the bird's foot. I took extensive precautions when working with the old museum samples to minimize contamination. In the lab, I extracted DNA from the samples, and then prepared that DNA for the sequencing that ultimately results in century-old genomes. Finally, we decoded the genomes using high-powered computers. From these genomes, we learned that some species experienced declines in their population size. Other species experienced increases in their population, and still other species maintained their population size. We have also learned about the distinct histories of connectivity in these species. Some species have maintained connectivity among populations amid habitat change, while populations of other species have become less connected. We used new technology to collect genomes from old specimens and learned how the changing environment has affected Ethiopian montane birds. This knowledge can help us inform conservation for not only these species, but global biodiversity.